So tonight we're going to be hearing from Amy, Technologies of Brain Control. Can't really get a more compelling title. Amy Smith-Dijak grew up on McGill University's agricultural campus in St. Anne de Bellevue, Quebec. She became interested in the brain and its dysfunction through interactions with autistic children in grade school. This interest was strengthened when she volunteered with Alzheimer's disease patients in St. Anne's Veterans Hospital. And she holds a Bachelor's of Neuroscience from McGill University. Right now she's pursuing her PhD in the university at UBC here. And her research focuses on synaptic scaling and Huntington's disease. And no, she does not work in the nice new Center for Brain Health. She works in the scary old psychiatry building behind it. That is the building on campus that is most likely to be haunted. And kind of a funny story. So when Amy was 2 years old, she could already know she'd be doing great things. So her parents asked her if she wanted a brother or sister when she was 2. She said, "No thanks, I'm too important." So, Alright, so join me in welcoming Amy.
Thank you very much. That was a very nice introduction from Doug. So yeah, I'm here to talk to you about technologies of brain control, which is what we see Professor X doing sometimes. Although I'm going to be talking about technologies that actually exist. They're fairly experimental to a greater or lesser degree, but they are things that are around and are being developed as we speak. So first I want to talk about some background of how we got to where we are and why we have these technologies. So I'm going to go back first to the 18th century, which is when people first started thinking that maybe there was some sort of electricity going on in the human and animal nervous system, which started with Luigi Galvani, who had a lab at the University of Parma in Italy. He was doing some experiments where he was dissecting frogs. He had his dissected frog attached to the table with a metal bar, and he was dissecting it with a metal scalpel, and the bar happened to be touching a nerve that was exposed. The scalpel touched the same nerve. Electric impulses went down that nerve, and the leg spasmed, which was surprising to him because as far as he knew, this frog was dead. And he did some more research to investigate this and decided that what this meant was that there was this animal energy that was animating all living things that was helping this frog to move, and that he had released this animal energy by cutting it. Then Alessandro Volta, another scientist, came along and he said, well, actually that's not right. You're getting this reaction because it's two different metals at each end of the nerve, and you're just, there's electricity flowing between them. And this was actually his inspiration to create the first battery. So we have frog legs to thank for the battery. The next step in this development comes in the 1800s with Lesion Studies, where basically what was going on is you'd have these physicians who were seeing patients. They would come in with various impairments in their functioning. Maybe they were having trouble with speech production. Maybe they were having trouble with moving their arm. This was after some sort of brain damage, usually a stroke. The brain that we have here is a very famous brain. It was from a patient seen by Dr. Paul Broca, and the area here where there's damage is known as Broca's area. And the patient that had this damage after his stroke was no longer able to speak. And when Paul Broca saw him, he characterized his deficits, and then when this patient died several years later, Paul Broca did an autopsy and looked at the area of his brain that was damaged. And so these doctors that were seeing these patients, they were talking to each other about these patients, and they realized that patients with damage in similar parts of their brain had similar deficits. Patients with damage to Broca's area couldn't speak. Patients with damage to Wernicke's area couldn't understand language. And this led to the idea that certain parts of the brain do certain things. Current research into plasticity has sort of changed the way we look at this a bit, but this is still a concept that underlies a lot of research today, and many fields have grown out of this, including neuropsychology, which is the study of the relation between parts of the brain and behavior, and less reputably phrenology, which is when people said, hey, if specific parts of the brain do specific things, then parts of the brain that are more dominant in people should be larger, and that should leave an impression on the skull, and therefore we can tell you about yourself by reading the bumps on your skull, which is now pretty discredited. So these lesion studies tell us certain things, but there's other things they can't tell us about. So in biology, we talk about things being necessary and sufficient for certain processes. When something is necessary for a process, that means that if it's not there, that process isn't going to happen. Lesion studies tell us whether brain regions are necessary for a behavior. Broca's area is necessary for speech production. Sufficiency means that an area of the brain, the activity in that area of the brain in and of itself is enough to produce a behavior. So if we activate Broca's area, does that lead to speech production? You can't tell that with lesion studies. You can only tell what happens when it's not working. You can't tell what's happening when it is working. So the first person to try to see what happens when you turn an area on, or one of the first people, was Wilder Penfield at the Montreal Neurological Institute in Montreal. He was a neurosurgeon, and when you do brain surgery, your patient is actually conscious for the entire time, so that they can report any unusual feelings that happen during surgery that are maybe indicative of things going wrong. And so Wilder Penfield decided, hey, while we have these patients in the operating room, why don't we try running an electric current through parts of their brain and seeing what happens? So if anybody is, and this was actually a fairly successful technique that allowed him to map the function of quite a lot of the surface area of the brain. If you've ever seen the Heritage Minute with the woman on the gurney talking about smelling burnt toast, I've talked to some people who think that she's having a stroke. She's actually not. That's Wilder Penfield stimulating her olfactory cortex and producing the smell of burnt toast. By the way, we actually still don't understand very well how the olfactory cortex works, even today. Two things that he was able to characterize very well were the somatosensory cortex, which is in yellow, and the motor cortex, which is in pink. So somatosensory cortex is where the touch information comes to your brain first when it comes in from the spinal cord. Motor cortex is where all of the motor commands for your muscles get sent before they go to the spinal cord. And he was able to use this technique to determine where sensations or motor commands for a specific part of the bodies were localized. So if he stimulated, say, the lip part of the somatosensory cortex, then the patient would feel a tingling sensation on their lips. If he stimulated, say, the leg portion of the motor cortex, the patient would kick. And this is a very useful piece of information that we still use to this day. The next step, which is something that's very close to my heart because it's very intimately related with what I do in my work, was in the late 40s and early 50s, Hodgkin and Huxley were the first people to record the electrical activity that was going on inside a neuron. So this was the first time that you were able to actually look at the activity of a specific brain cell. They were benefiting a lot from the technology, from the communication technologies coming out of World War II. Back then they had to actually physically put an electrode inside the cells. So they used these squid giant axons that have a millimeter wide axon, which is humongous in biology, and they recorded the pattern of electrical activity that was happening in the cell when they stimulated it. And it's a really beautiful piece of work, and if you're interested in biological modeling, you should check it out because it's awesome. So what is an action potential? This is important for some of the stuff that I'm going to tell you about. So just a brief overview, we have our neuron here, our brain cell, and there's a cell body, and there's all this things coming off of it. These are called dendrites. They are contacted by other brain cells, and those brain cells release neurotransmitters onto them that causes a signal to run down the dendrites to the cell body. Some of those signals tell the cell that it should fire an action potential. Some of them tell them that it shouldn't. They all vote on it. It adds up their votes, and then based on that, it decides to fire or not fire an action potential. And if it decides to fire an action potential, then a pulse of electricity goes down the axon here, which then splits up and makes contact with other cells, and the cycle continues onto the next cell. So this leads us to our first method of brain control, which is classical stimulation. This was sort of related to what Wilder Penfield was doing, but more in depth. So in this, you stick an electrode into the brain and you put electricity through it, and that, because your brain cells work on electrical impulses, if you inject current into them, then that will change how they behave. Depending on how you put it in, you can inject current into a large structure in the brain, or you can inject it into a single cell. This has been a really useful technique in the past. We've learned a lot from it, but it's not ideal. Especially if you're doing large structures, you can't control what types of cells you're affecting, which is a problem because different structures have many different types of cells, some of which are working against each other. Also, as I said, you have these axons that are having these electrical impulses going through them, and there are axons that are going through your structure that you're stimulating from one completely different part of the brain to another completely different part of the brain. So it tells you something, but it's very difficult to do a very sophisticated interpretation of the results. Also, there's a lot of very subtle biochemical things that are involved in brain transmission, and when you're just injecting current into them, you're sort of bypassing a lot of these. Is potentially not a very naturalistic approach to brain science. And also, because this is very invasive, you can't do this with healthy participants. You can only do this in animal models or people who already have electrodes implanted because they're waiting for brain surgery. We've done a lot of this work in epilepsy patients who are waiting to get the area that's causing their epileptic seizures removed. But of course, your problem is if you're only looking at epileptic patients, is your conclusion about how the brain works, how the average brain works, or is it how epileptic patients' brains work? That being said, there's some really, we've had some really interesting findings from this, one of which that's kind of a classic was regarding the source of pleasure in the brain. So you may have heard that dopamine is the pleasure molecule. This all started back in the 50s when there were a couple of researchers who were trying to see if actually the septal area over here was involved in learning. So they put electrodes into mice's brains, into that area, and they put the mice in a box like this one and would inject current into this area when the mouse reached a certain area of the box, and their idea was that this would cause the mice to avoid that area. It didn't work, but there was this one mouse that when it reached the area where it got stimulation, it was just incredibly, it never wanted to leave. It would just stay in that area indefinitely, getting stimulation for as long as it possibly could, which was very different from the other mice and very different from what they were expecting. And they couldn't figure out why this one mouse was like this, and they couldn't just, open up its brain and see where the electrode was, because maybe no other mouse was going to react like this. So they actually built a small lead jacket for this mouse and took it to the X-ray department and had it X-rayed to figure out what had happened with this electrode. And it turned out that the electrode was not in the septum over here. It was halfway across the brain in the midbrain. And that's what it was stimulating. And it turned out, and from this, they figured out that there was these circuits involved in the midbrain that were involved in pleasure, and there was dopamine released in these circuits, and it started a whole new field, and it's important in addictions research, and all because one electrode didn't go into where it was supposed to go. Another really cool experiment that was done recently last year by Parvizi et al. Was a case study in 2 epileptic patients where they were using the electrodes that were already implanted for preparation for surgery. They went to the patients and said, can we please, you know, borrow your electrodes for a little while so we can do our experiments. And these electrodes happened to be placed in an area that they were interested in, so they took these electrodes, they ran current through this area, and they found that when they stimulated this area, they actually got a pretty complex response that was very similar in both of them, which they labeled the will to persevere. Are you like interested in the brain region? Oh yeah, sorry, yeah. Yeah, so there is a video here of the patient recording what they have. Unfortunately, the sound isn't good. I don't know if you guys are interested in that.
Yeah, my upper respiratory started kind of, my chest and respiratory system started getting kind of shaky, like it was wanting to push itself out the door.
Really? Okay. Okay. Did you feel any change in your emotion and mood?
Well, not my emotion, but in my mood, I started getting this feeling like I was driving into a storm. That's the kind of feeling I got. It was almost like you're headed towards this storm that's on the other side of maybe a couple of miles away, and you got to get across the hill. And all of a sudden, you're sitting there going, "How am I going to figure out how to get over that, through that?" And that's the way my brain started functioning.
So, stimulating this area produces this very complex emotional memory response, and it was pretty consistent across these two patients, so the authors were using this as an argument that this particular area is involved in this will to persevere. This is obviously a very invasive thing, but there are some people who are walking around with these electrodes in their brain because there is a thing called deep brain stimulation, which is very effective in some Parkinson's disease patients, where what you do is you place an electrode into the brain in the Parkinson's disease patients in a specific area, and you put a little stimulator into their chest there that controls the rate of electrical impulses that go into that area, and then you turn it on and you let it go, and they actually don't know why this works, but it really decreases the Parkinson's disease symptoms in patients that aren't responding well to medication. And it's also in trials for other disorders, including depression, Alzheimer's, OCD. But yeah, we have no idea why this works, but it's an example of this very old technology of brain stimulation actually having, you know, some real-life clinical applications. Yeah, so before I talk about the next technology, I just want to do one more, explain one more thing about electrical transmission in the brain. So basically, the electrical signals in the brain are controlled by the flow of charged atoms called ions across the cell membrane. They flow across the cell membrane through these proteins that have holes in them that let through only specific ions, and they can be open or closed depending on the characteristics of the cell, how it's feeling at the moment. And these proteins are called ion channels. And as I said, different types of signals make it more or less probable that the cell will fire an action potential. And basically what you need to remember is if sodium is flowing across the channel, that if there's more sodium flowing across the membrane, then that increases the probability that the cell will fire. And if potassium and chloride or chloride are flowing across the membrane, then that decreases the probability that they're going to fire. And this is related to optogenetics, which is what I just spent the entire day trying to get to work today. And the basis of optogenetics is that there are some bacteria that have ion channels which will open in response to specific wavelengths of light. So our very helpful molecular biologist friends have managed to clone these genes and improve them a bit so that they're more suitable for research, and then we are able to take these genes and put them into the cells that we're interested in, and then we get these ion channels in our membrane, and when we shine light on the cells, it changes the electrical properties of the cell and changes how they work, which is very, very nice for those of us trying to, test the effects of cell firing without just shoving a whole bunch of current into our cells. Something cool that was done with this, paper published last year by Ramirez et al., where they created a false memory in a mouse using optogenetics. They put channels into the cells that were activated when the mouse was in a specific area, and only those cells. And then they activated these cells while the mouse was not in that area but was having its foot shocked, which is a fear conditioning paradigm. And then later, when they placed the mouse back in that area, the mouse was afraid. Even though nothing bad had ever actually happened to it in that area, they had made this false memory that something bad had happened to it in that area. And when I tell people things about this, they say that this sounds pretty evil, which it does. So what are the non-evil applications of this kind of thing? So for research, it's got some really really promising possibilities that are very exciting to people like me who do basic research, where, as I said, with neurostimulation, when you just put an electrode into some brain area, you can't actually control what types of cells you're activating. But with this, you can. You can say, I only want these channels in this type of cell, and then you shine a light on, and only that type of cell responds, which is pretty great. And also, you're activating them in a more naturalistic way than when you're just injecting current, because you are opening the ion channels, which is what usually happens when the electrical activity of these cells changes. So that's also, it's a little bit closer to what's actually happening. And there's also potential in the clinic, in psychiatric disorders. For example, that paper I was showing you created a false memory in mice, but it's also possible, there's also been papers that have erased a memory in mice. So if we could use this on patients with PTSD to erase or dampen the memory that's giving them PTSD, that would be pretty great. Obviously, this is something that's potential for abuse, so you want to be careful about that. Also, certain neurological disorders could benefit from this because stimulation of the degenerating neurons with certain patterns could help to make them more healthy or even to stop them dying. So that would be nice too, and non-evil. The last thing that I want to tell you about, which is the magnets part of my title, is transcranial magnetic stimulation. The main advantage of this technology over stimulation with an electrode is that it is completely non-invasive. What you do is you have a large magnet that creates a very focused magnetic field over a very specific, over a targeted part of the brain. You can focus it to an area down to an area of about 1 centimeter cubed, which is not bad. And you can use this to switch this to excite the activity in this area or to inhibit it. If you're sending more than 1 pulse per second, it usually excites. If you're sending less than 1 pulse per second, it usually inhibits. Where this is a pretty new technology. The earliest papers I've seen about it are from the 90s. So we're still kind of in a characterization phase where we're still figuring out how it works to a greater or lesser extent. But there is some practical applications being researched already. For example, there's a group at UBC and in other places as well that are looking at this for recovery after brain injury, specifically after stroke. It's been found that when you do rehab for stroke patients, if you stimulate certain relevant areas during rehab, you get better recovery. So it would be nice if you could use this to help stroke patients recover or other people with other brain injuries. Oh yeah, so I'm going to show you an example of this now, but So I'm going to show you an example of a study that was using this to investigate a specific illusion, but before I show you the study, I want to show you the illusion. It's called the McGurk effect, as you can see.
Ba 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 ba.
Three different videos that were actually played with the same sound, but if this effect worked, which it usually does, then you would have heard different syllables when different videos were played because your visual information is overriding your auditory information. So there was a group of researchers, Beauchamp et al., that a few years ago they look at this specific area called the superior temporal sulcus. That's an area of multisensory integration, which the McGurk effect is an example of because it's auditory and visual information interacting. And so they thought, hey, maybe this area is involved with this. So they used TMS to inhibit that area, and they found that when they inhibited this area, the perception of the McGurk effect went down from 90-something% down to 40-something%. So that's pretty good evidence that it is involved. So, as I've mentioned, people often say that these technologies sound a little bit evil. So what is the potential for misuse of these technologies? In the case of neurostimulation, this isn't something that's going to be used in some widespread manner because it does involve brain surgery to implant the electrode into the person. This requires a lot of effort. Also, as I said, we don't really know how deep brain stimulation works, so it would be difficult to produce any sort of predictable, consistent effect with stimulation. However, one thing that is of concern, it's been found recently that you have people walking around with neurostimulators in their brains because they are undergoing deep brain stimulation, and it's been found recently that you can hack into medical devices. So if somebody wanted to, they could hack into deep brain stimulators. As I've said, because we don't really understand the mechanisms, it would be difficult to produce any sort of complex behavior with this, but you certainly could give them a seizure or something like that, which could be very damaging. Optogenetics, maybe Men in Black's memory wipe is not so far away. The thing that's a little bit scary about optogenetics is that the way that we usually insert these channels into mice is with a virus, so it wouldn't be that difficult to insert these channels into a fairly wide population. That being said, it's not difficult to get the channels in. It is difficult to stimulate them because they have to be stimulated by light that is if you're going to stimulate them without having a light implanted into the area that you're trying to stimulate, then that light has to go through your hair, it has to go through your skin, it has to go through your skull, and it has to go through whatever brain tissue is between your skull and the area of interest. Thus far, there is no channel that is able to do this. There was a recently published paper about touting this great advance in optogenetics where they could stimulate channels that were a whole 3 millimeters under the surface of the brain, so long as the brain was exposed and there was no bone or skin. So we're not really there yet. That being said, if someone were to develop a channel like that, that would be very worrying. But currently, controlling somebody's brain by optogenetics would have similar drawbacks to controlling it by deep brain stimulation, except that there's actually nobody walking around with optogenetics in their head with the optogenetics and a light in their head that I know of. And finally, TMS. This is probably, again, not going to be misused on a population level because you have to have a magnet at a pretty consistent distance from their heads, so you can't really do that to a large crowd or something like that. That being said, this is something that one could see potentially being used in, say, an interrogation setting, if you were trying to make somebody less inhibited in order to tell you information. Also, perhaps somebody might try some sort of brainwashing with it. That being said, like I said, as I mentioned, we are still in a characterization phase with this, so if you were trying to brainwash somebody with this, it would be very difficult to get a predictable result with that. And if anybody's planning on brainwashing anybody, I would not recommend using TMS. So should we be worried about this? Like I say, probably the thing that's most worrying at the moment would be somebody hacking into deep brain stimulation implants and using those to potentially really damage somebody if they wanted to. Optogenetics could be of concern in the future because it would be not particularly difficult to spread these channels around, but as they say, that until we get the technology where somebody can actually stimulate those channels once they're inside your head without surgery, it's probably not that likely that they're going to be, it's probably not that concerning. And TMS is probably the one that's least likely to be misused on random people, just because if somebody's holding a magnet at a very specific distance from your head, you're probably going to notice. But the potential for misuse in interrogation is it's something you could see happening. So are there questions?